Hello friends, welcome to another video tutorial from Shomu's Biology. In this video lecture, we'll be talking about the activation and maturation of T cells. Not the activation, because we talked about the activation of T cells during the immune response and how the immune response gets activated due to the engagement of T cells with the antigen presenting cells. But here we will talk about the training of T cells, because T cells' prime job is to get in contact with the cell mediated immunity as well as the humoral immunity, and they achieve that by engaging in interaction with the antigen presenting cell. And during the engagement with antigen presenting cell, they need to find out whether the molecule that is provided by the MHC class 2 is a self molecule or a non-self molecule. So T cell have this ability to figure out whether a fragment of protein is a self or a non-self. Now, how would they find that? That is the most important thing. To find that out, they need to get trained in a specific region in our body known as thymus. So all the cells that are produced, known as thymocytes, which are at the earlier verge of their life, they have many receptors on their surface. They can recognize both self, both non-self, though all they can engage in interaction with all these things. Among all those populations, we need to figure out which cells are fitting best for the desire of our immune system, that is, to distinguish between self and non-self. Now, those cells who fail to distinguish between self and non-self are getting killed with the process called apoptosis or programmed cell death. And the cells who can perfectly recognize specific pathogen and also recognize the self molecule and exclude that, those cells will be selected and they will be taken for the next journey for the activation from naive T cell to either CD4 cells or CD8 T cells. And those T cells will have different functionality and they will be in field of immunity to protect us. So let's look at here in this whole process of T cell maturation. For this, we need to understand a simple thing about the whole process of maturation is taking place in thymus. Now, if I draw the cross section of thymus, thymus have different regions. For example, this is the subcapsular region, then they have a cortex, then they have a corticomedullary junction, then they finally have a medulla. So they have all this region. This is medulla, corticomedullary junction, cortex, and I must write it from here. Medulla, corticomedullary junction, cortex, and this is the subcapsular region. This is the kind of, I'm not interested in talking about anatomy of this region, but this is how it looks like. Now the thing is, this whole thing is medulla, for example. Now those thymocytes take entry into this thymus with the help of those venules that are present in medulla or in corticomedullary junction. The venules are present. The high endothelial venule is one such type of venule through which those thymocytes start taking entry into this thymus, and actually they, after taking entry, they mostly present in the subcapsular region. All these are the thymocytes. Thymocytes are the precursor type of cells which will be developed into T cells later. So thymocytes are present now mostly in the subcapsular region. Now as they start getting matured, they will come down towards the medulla. So they make their journey from subcapsular region towards medulla, like the semesters of their education system. 4 semesters are there finally to get graduate and then leave this thymus to ultimately reach the lymphatics as well as the bloodstream to fight against the infection. So it's just like a graduation course they're doing in there. So what the figure and what's the features that we need to look for? All these thymocytes at this point of time, all of them have both CD4 as well as CD8 receptors on their surface. They have CD4 receptors, they have CD8 receptors, they can bind with both MHC class 2, they can also bind with MHC class 1. But we know specialized cells, we have to produce specialized T cells that some interacts with MHC class 1, some interact with MHC class 2. So how it will take place? Actually, there are 2 types of selection procedure going on there. First is the selection procedure based on whether the separation, whether it will be CD8 cell or CD4 cell. That separation is mostly carried out in the subcapsular region and in a little bit of the cortex. Okay, and there is a second process known as the actual selection process, or known as the thymic selection process. The thymic selection process will be carried out mostly in the corticomedullary junction and medulla. So there are these two different processes going on. Let's talk about the first process that we discussed here. The first type of process to find out and distinguish between CD4 or CD8. So So what we will see here is that there are subcapsular cells, there are epithelial cells, subcapsular epithelial cells that are present in this subcapsular region. Now those epithelial cells, they will showcase, let me explain, let's say this is the subcapsular epithelial cell, and they start expressing either MHC2 or they can also start expressing MHC1. They can express any of that, and these thymocytes can engage in the interaction. Now those thymocytes who engage in the interaction with MHC2 and properly understand the interaction with MHC2, those are selected. Let's say this is the thymocytes who engage with MHC2 in a proper perfect interaction, a proper avidity binding, not too heavy or not too less. Now those who won't be able to, like in this case this T cell is properly binding with this MHC2, finding that. So this T cell will be selected and its T cell receptor will be present there, but the CD, so once they will interact with MHC2, the CD4 receptor will be kept, while the CD8 receptor will be degraded, because they will not need CD8 anymore, because we are differentiating between CD4 cells and CD8 cells. So once they engage properly with MHC2, they will only select CD4, CD8 will be destroyed and those cells will be selected. So now those cells will be only CD4 cells. Similarly, if some of the thymocyte cells engage in interaction with MHC class 1 in proper engagement, in that sense they will be selected for CD8 and the CD4 will be destroyed. So CD4 will be destroyed, CD8 will be selected to grow, and that cell will be selected for becoming CD8 T cell or cytotoxic T cell, while the other cells we talked about were selected for CD4 will be known as T helper cells. That's how they differentiate between CD4 and CD8. Now let's say some of those cells cannot bind with either MHC1 or MHC2. They fail to engage in proper interaction with either MHC1 and MHC2. Those cells will be signaled for apoptosis or programmed cell death. Or some of the cells are there who can bind with both MHC1 as well as MHC2. So we have one type of cell binds with MHC2 which is selected, another type binds with MHC1 which is selected, third type which binds with none, which will be destroyed by apoptosis, and the fourth one who binds with both, again destroyed by apoptosis, because we don't want them, they will create mess, because if they bind with both of them and too eagerly, that will be also dangerous. We need optimum things, and that's what we selected both of them earlier. Okay, so that's how they trained and differentiate between CD8 and CD4 cells in the subcapsular region there, and it is guided by this subcapsular epithelial cells. Now once they are trained here, they start migrating towards the cortex and the corticomedullary junction and this area, and then finally to the medulla region. There comes other cells, there comes the macrophages, because this is the area where macrophages are present, B cells are present, all these other type of body own cells are present. Now here comes another final test, the final exam to become graduate, and that will be very difficult, and that is the major exam to find out whether the macrophage or those dendritic cells or whatever cells that are present here, macrophages, those B cells, those are self or foreign. They need to figure that out now. Okay, now what will happen, let me tell you here. Now let's say this is, let's say, a body cell, our own body cell. Let me draw it with a green color here. This is our body cell. So here at the end we have those body cells like dendritic cells or macrophages over there. Now they are representing some part of our body's own self protein molecules on the surface, and now T cells final test to find that out and bind with it. Now again there are three different responses that can be done. That one is that T cell is not at all engaging with any interaction with it. That means T cell is not recognizing the self particle. If T cell fails in interaction or fails in recognizing the self molecule, in that case those T cells will be destroyed by apoptosis. Now the second scenario, when again the self particle, I mean those, let's say the antigenic those dendritic cells for example, showcasing their self material outside, and again it comes another T cell. Now this T cell is properly interacting and selecting and identifying the self molecule and engage in a little interaction there, but this T cell is not reactive, it's not releasing any chemokines or anything of that kind. So it's not reactive, but it can identify the self molecule, it can adhere to the self molecule. That means this T cell's training is successful, its training is complete, that it can engage in attachment with the self molecule, it can understand what is self or not. So it is passed the exam. So now this cell can be released from this medulla through the high endothelial venules to the venules to the bloodstream or to the lymphatics. Those cells are selected. Now there is a third kind of reaction which is also possible. Now let's say again self proteins are like placed, and now the T cell comes in, and this T cell is recognizing this self peptide, that's a good thing, but they are interacting with so avidly and start responding against that, starts releasing chemical factors to kill that. This is wrong, because they are super active in this sense, they can't recognize it as a self. So even though it recognizes very tightly and hold the self molecule as pathogen and start doing those pathogenic responses against it, try to kill it. So this is again horrible thing. So if we make this T cell pass, this T cell will start destroying our self immunity and it can cause an autoimmune disorder. So cells idea is not to keep them alive, to again kill them with the process of apoptosis or programmed cell death. So again they will be also taken for the process of apoptosis. So only those T cells are selected who are interacting with the target or self peptide molecules with a normal affinity and not responding to that much eagerly. That is the T cell, those are the T cells which are selected. Now remember, the whole process begins between the differentiation of CD4 and CD8 T cells, start with this subcapsular region, then the maturation actually take place in the cortex. I haven't drawn that even, but this whole process of selection and maturation, these things take place in the cortex. Now once they start this maturation process, those cells are coming down ultimately to the medulla, where they finally receives and interacting with those antigen presenting cells like dendritic cells or these thymic macrophages that are present there. Upon involving with this interaction, they are finally trained, and those trained T cells will be transferred through these venules to ultimately to the bloodstream, and they can receive and go to those tissue layers, and they can fight against the infection, or they can also be transported to the lymphatics for storing there for a while. That is the overall idea of the T cell maturation and differentiation process. And this whole thing is responsible by all those thymic cells, as well as there are some chemical signaling going on which we haven't talked about much yet, because there are some sort of signaling also required in this sense to get them activated. Once they are active, they will be moved there, they will be moving towards the infected site by the chemotaxis mechanism, and once they reach there, then the final process of their final activation, they will be activated by engaging interaction with macrophages or dendritic cells which are present there, and then they will call upon many more T cells and many more other B cells and stuff to finally activate the cell mediated response, as well as they will convert B cells into plasma cells to make antibody, which is also communicating with the humoral mode of immune response. So in that sense, this whole process are linked. The only reason I keep repeating the stuff, I keep repeating what we learned earlier, what we talked earlier, is to make this connection strong enough, because immunology is a huge thing and it's all about connecting the dots in immunology. If you fail to connect the dot in immunology, you will never understand immunology well. So it's very important idea to get a big picture very clear about all this process. So that's all about the T cell mediated response and T cell activation and maturation. I hope you understand the video. If you like this video, please hit the like button, subscribe to my channel to get more videos like that, and definitely share this video with your friends. Thank you.